going to talk about aspects of transplantation. And so that's where we're going right now. So this figure shows you the number of transplants performed in 2016 for some selected organs. So what you should see here are these numbers to give you an idea of the frequency of what we're talking about here. So you can see, heart transplants are happening in about, about 3,000 per year, 19,000 kidney transplants, and so on. The organs that are shown on this figure are all examples of one of two major types of transplant that we're going to talk about. Really, I would say that there are three major types of transplant. All of these are kind of examples of one of them, which is going to be the majority of what I'm going to talk about today, although I will hit two other types at different times. These are known as solid organ transplants. And so in general, when we talk about transplantation, we often talk about solid organ transplants. Most of the time, when I give you examples of transplantation, I'm probably going to give you examples of kidney transplants. You can see that kidney transplants are the most frequent, and a big reason for that is that we all have two kidneys, and so we can actually act as living donors, and so the number of kidneys available for transplant is a bit different than some of the other organs. So I'll probably often be like, when we're transplanting a kidney, blah, blah, blah. It could be any of these solid organs. The other two types of transplantation that exist are blood transfusion and bone marrow transplantation. And so those both have some slight differences from solid organ transplants, and we'll get into the details of each of those a little bit as we go forward. So we also I want to just sort of think a little bit about transplantation and why some of this is important. So this is sort of an image that is sort of depicting a transplant scenario. So we've got some patient who has some type of usually chronic disease where they are having a problem with a particular organ, and in some cases they may die without that organ. And that patient is usually pretty sick and is spending a fair amount of time waiting for that organ. We'll have a situation where an organ becomes available for transplant. And that is a relatively rare situation. As I'll say more about at the end, we have far more patients who need organs than organs available. And so we've got sort of this very rare scenario where there is an organ available for a patient who needs that transplant. We are likely going to be doing a whole lot of very sophisticated work to get that organ to our patient quickly, so that organ is still viable. We don't want to have that organ start to undergo a lot of cell death. We don't want to, basically we want to have the organ still be fresh enough that it will work in our patient into whom we are transplanting. So we might be, flying organs around and doing a whole bunch of very sophisticated work. The patient is going to get called in the middle of the night. You can see them, they're getting a phone call in the middle of the night. The doctors are getting called in the middle of the night. Everybody's going to rush to the hospital, start this very complex surgery. And the problem that can happen immunologically is that in some cases, when we're transplanting in that organ, we can have an immune response to that organ, and your immune system can destroy that organ. And so all of that hard work that happened at the beginning will be for nothing. And our patient will be kind of back to where they started from. And so understanding the immunology of what's going on here, both so we can prevent these types of things or figure out ways to treat them, is quite important. Much of this was described, or some of this was described with original experiments with mice and with giving mice skin transplants, so moving skin from one mouse to another. So a lot of the general details of transplantation often come into these mouse skin experiments. You will see on this slide that we use the word graft. And typically when we talk about transplantation, we will talk about grafts a lot. So we might talk about the graft, which is just another word for the thing that's getting transplanted. So if we had a kidney transplant, it would be a kidney graft. We'll also often talk about the host, that's whoever's receiving the transplant. There are a number of different prefixes that come up when we think about doing transplants. Sometimes we'll put them in front of graft. I'll go through some of those in a couple of slides. But just so be aware that's what graft is. And people can do some experiments like the experiment shown here. And so we could first have a mouse of strain B, and we could give that mouse a skin graft from strain A. So you can see the skin doesn't quite match the mouse. If we do that, if we give a mouse a non-matched skin graft, within about 14 days, we will see that skin graft get rejected. And so you can see that rejection. So in fact, that skin is going to get attacked. It's not going to persist in that mouse. If we then were to give that same mouse another skin graft of type A again, which is what you can see happening on the top here. So we're giving the mouse a second skin graft of type A. That second graft will again be rejected. And that rejection will happen more quickly. Now it might be happening in 6 days as opposed to 14 that happened before. And so if you just look at that process that's in the top row on this slide, what might you infer about transplant rejection? Yep, Jamie.
There's memory involved.
There's memory involved. Or more specifically, this must be an adaptive immune response because it changes upon a second exposure to that similar graft. And so this starts to say, okay, something about what's going on with these transplants and with transplant rejection involves the adaptive immune system. We can also do experiments like the one that's shown on the bottom, where we take our mouse who has rejected its skin graft and we isolate T cells from that mouse. We can give those T cells to a naive mouse and then give that mouse a skin graft of type A. And that mouse will also reject the graft quickly within about 6 days. And so what might you infer from those data? Yep, Sebastian.
It's the T cells that are causing the adaptive response.
And so this also will tell you that perhaps T cells are mediating that rejection. One of the clearest types or the most famous types of rejection is T cell mediated. There are some situations where it's not T cell mediated, but the sort of classic rejection response is a T cell mediated response. And so we see this sort of hallmark of the adaptive response as we're looking at rejection. You can see this here as well. So basically you can see what you would want to have happen is you would want your skin that you're putting in to get new blood vessels to grow in and to heal and to live happily ever after. In that first situation, you're going to put in your skin, you're going to get some blood vessels, but then you're going to get inflammation and you're going to actually destroy. And that's happening in 2 weeks in that first case. In the second case, you can see that's happening much quicker. This slide also mentioned, uses a couple of terms, and so I'll just give you those terms right now. So this slide mentions the term autograft. And the alternatives to autografts are allografts or xenografts. In general, the prefixes auto, allo, and xeno are used a lot when we talk about transplantation. So sometimes I'll think about like an auto reaction. Sometimes I might talk about something that's called an autogeneic or an allogeneic response. These are all coming from the same place. So an autograft, which is what it lists on the left side of this slide, is a graft or a transplant from someone who is genetically identical to you. Usually, most of us are not lucky enough to have someone who is genetically identical to us in terms of like a twin. If we're in mice, that's kind of easy because mice are inbred and genetically identical. Most often when we see autografts happening, they're actually grafts from you to yourself. So if, for example, you have an issue with your skin, you might actually get skin from another part of your body transplanted. You might, if you were going to have a procedure done that might impact your immune system, you might have bone marrow harvested beforehand so you can get your own personal bone marrow back. You might have blood taken that is then even given back to you. And so all of those would be examples of autografts. The idea with an autograft is you're going in from the host and the donor and the recipient are genetically identical. And you are genetically identical to yourself. If you were in another situation where there were two genetically identical people, or we were in mice, those would also be autografts. Allografts, or allogeneic reactions are actually very frequently what we think about a lot when we think about transplantation. Allo means that we are getting a transplant from one member of a species to another, but they are not genetically identical. So this means you're getting something from another human, but they're not genetically identical to you. And so sometimes we'll talk about allografts or allogeneic responses or alloreactivity or things like that. That basically means you're getting something from another human, they're not genetically identical to you. We also have xenografts. Xenografts are where you're getting a transplant from another species. And so those are kind of the three big topics here that come up. When we think about transplant rejection, there are three major types of transplant rejection. There are kind of three big processes that are important, especially in solid organ transplantation. Those three are known as hyperacute rejection, acute rejection, and chronic rejection. And I've listed them there based on timing. So hyperacute happens first, acute happens second, chronic happens third. Those three types of rejection can map onto the types of responses we saw last time with hypersensitivity. So hyperacute rejection has a lot in common with type 2 hypersensitivity. Acute rejection has a lot in common with type 4 hypersensitivity. Chronic rejection has a lot in common with type 3 hypersensitivity. So we're going to be seeing a lot of the same themes that we have already seen. We're going to talk through these three types of solid organ transplant issues with some in time order now. So the one that happens first, if I was going to give you a transplant, would be hyperacute rejection. As you can see on this slide, hyperacute rejection has some similarities to type 2 hypersensitivity response. So based on that, what do you know about how hyperacute rejection works? What would you say is how hyperacute rejection works? What kinds of things should it involve? Yep, James.
IgG or IgM antibodies.
So we're going to have IgG or IgM antibodies, and what's the other thing that's going to be really key? Aaron.
The antigen is on the cell surface.
Antigens on the cell surface. And in fact, both of those things are true in hyperacute rejection. So in hyperacute rejection, antibodies are key. One of the things that is important with the antibodies in hyperacute rejection, in fact, it is also true that the antigens are going to be cell surface antigens. So all of that is, everything you just said, 100% true for hyperacute rejection. The key thing about the antibodies in hyperacute rejection is that these antibodies were pre-existing. So these are antibodies that you already had. You didn't have to make those antibodies. The antibodies were already there. That means those antibodies can bind to the cell surface antigen right away, as soon as you get that transplant. And we can start to have things like complement-mediated lysis, we can have phagocytic cells, we can have inflammation, and we're going to destroy that graft, in this case, a kidney. And so this is going to happen, within perhaps a couple weeks after our transplant. And so you can see this here as well. Here's our patient. They have received a new kidney. It's been surgically put into their vasculature. However, sadly, they have pre-existing antibodies that already bound to some antigen in this kidney. Strangely, they are shockingly large for the size of this person. Those antibodies will bind to those antigens that are on the surface of the vessels in that kidney, start to destroy the vessels. When the vessels are destroyed, the kidney can't get blood flow anymore, and so the kidney will actually die. Yep.
How long does it take the immune system from the time it starts attacking the organ to stop the organ from functioning?
So in hyperacute, it's going to be a couple weeks.
Is that a couple weeks for it to activate, or a couple weeks for organ?
No, it's probably going to be a couple weeks-ish for the organ to be completely destroyed. Because you don't need to wait for B cells to start making those antibodies. Key thing here, antibodies are pre-existing. So now we can also take a step back and think about this again. The idea here is that antibodies are pre-existing. If you think about it, in general, do you right now have a reason to have antibodies against my kidneys? Like, you did not evolve to attack other people's kidneys, right? Like, that's not what your immune system evolved for. Other people's kidneys were not the biggest threat in evolution. So the question that might come up here is, why the heck are there these pre-existing antibodies that are going to be an issue here? How the heck do you have pre-existing antibodies to other people's organs? And it turns out there is a very important and a very common antigen. That's the problem here. It is theoretically possible there could be another antigen. I'm not going to say there has never in the history of the world been another antigen. I'm sure there has, but there's one that's usually the problem here. And the antigen that's usually the problem are blood type antigens. So remember that in a person with type A blood, that person has antibodies against the B carbohydrate. A person with type B blood has antibodies against the A carbohydrate. Person with AB has doesn't have any antibodies. A person with type O blood has antibodies against A and B. You can actually see that better here on the left, where you can see each type and what antibodies they have. At the top, you can see the cells and which antigens they have. That limits your possibilities for transplantation. A person with type O blood can only receive type O blood cells because they have antibodies to the antigens present on everybody else's cells. Type A can only receive O or A because they have antibodies. Type A can only receive, type B can only receive O or B. AB can receive anything. And so most commonly the pre-existing antibodies that we see here are from blood group mismatches, where you have antibodies against some of these carbohydrates. As I've mentioned before, these carbohydrates are sort of present on all microbes in the GI tract, and so you're going to prime these B cells if they have not been killed by central tolerance mechanisms. This also, of course, plays a role in the other type of transplantation, blood transfusions, as I mentioned. And here you can also see so the general information about transfusions. When you're transfusing an organ, that organ is going to have some residual blood. You also are going to have those blood group carbohydrates on some parts of the organ, and so that's where those pre-existing antibodies are going to be attacking. Again, this also comes into the blood transfusion. You can see this as well as with the Rh, positive and negative, in terms of how we can have donors and recipients in blood transfusions here. The same rules apply when we're thinking about organ transplantation, and I always show this slide because everyone finds it very interesting to see the frequencies, if they know their blood type, to know if their blood type is rare or common. So for example, I figured out from one of our blood typing kits that I was B positive, so I'm kind of like medium in terms of how rare or common that blood type is. So generally with hyperacute rejection, the biggest thing, the big problem is these pre-existing antibodies. And in general, the pre-existing antibodies that we're going to have problems with are antibodies against ABO blood group antigens. So if you became a transplant surgeon, how would you deal with hyperacute rejection? How would you try to make sure that your patients did not have blood group rejection? Yeah, Justin.
Could you suppress their immune system?
Maybe you could suppress their immune system. Suppressing their immune system, it can be hard and can have some dangers. So there's something else you might try first.
I would preventatively test the organs or test each person's blood type.
Yeah, you basically would just test and only do transplants where you're going to not have to deal with this problem. And so you're going to do testing, you're going to get around hyperacute rejection by blood typing to make sure that this isn't a problem. Yep.
If a person is showing signs, is it a way to reverse it or take the organ out and save it, or once there's rejection?
You're probably not going to be able to take the organ out and save it. That organ is not going to be surviving outside of the body for a very long time. It's already been outside the body for a while, so taking it out a second time is not going to be healthy for that organ. When we get to acute rejection, which is mediated by T cells, we're going to talk more about suppressing the immune response and T cells. Here, because it's pre-existing antibodies, you would need to kind of be suppressing complement, and you need to be suppressing macrophages, and that's a lot harder to do body-wide What you can do sometimes is plasmapherese and try to remove some of those antibodies, but it's probably not going to be very effective. And so most likely in that case, you're going to lose the graft, and you're going to have to retransplant that patient, which is going to be terrible. And to be perfectly honest with you, most of the time right now, at least in the United States, when hyperacute rejection happens, the next thing that happens is a gigantic lawsuit to the hospital for not doing blood typing, because blood typing takes about 5 minutes. Yeah.
Sorry, this is a very specific question.
Go for it.
But is there a potential for CAR T cells to be used in this case to stop the B cells that are creating those antibodies?
Maybe, but the problem is the antibodies are already present.
Okay.
And so maybe you come up with a way to stop B cells from making those antibodies, but you're not going to you're unlikely to get to a situation. Maybe if you treat all infants immediately upon birth with those T cells, so the B cells never get activated.
I don't know, it sounds like a bad idea.
Maybe that would work, but it takes 5 minutes and costs almost nothing. Blood type.
Yeah.
Yeah.
If you give an Rh positive host an Rh negative organ, does the organ have any antibodies against Rh in it, or are they just not significant compared to the rest of the bodies?
They might have a few. It probably won't be significant compared to the rest of the body. So that probably won't be a huge issue.
So like that wouldn't cause rejection?
That probably wouldn't cause a ton. Well, so in that case, you're not going to attack the graft. You're going to attack those antibodies are going to go into circulation and attack the host. It's probably going to be few enough that you're not going to see a whole lot of damage. Yep. What did you say? No? Okay. Other questions? That will relate to something else we talk about later, Aaron. So it's a good question, just not quite yet. All right, so that was hyperacute rejection. The big type of rejection that is really important here is acute rejection. Acute rejection is really the one that people think of when they think about transplant rejection. So acute rejection has similarities to a type 4 hypersensitivity response. So based on that, what do you know about acute rejection? Addie.
T cells are going to cause the problems.
This one's T cell mediated. So if you remember when I showed you those mouse experiments earlier, we were transplanting T cells, and T cells were important because it was an acute rejection situation. And again, acute rejection is really the rejection that people tend to think about the most frequently. Acute rejection is T cell mediated. And so you can see the details of acute rejection here. So with acute rejection, we will put in some type of organ, some kidney, for example. That kidney will have some antigens. Here it's showing dendritic cells. We'll get into the details of the antigens in a couple of future slides. So right now, all you care about is there's mystery antigen. Those antigens are going to go to the nearby lymph node. We're going to turn on T cells. Those T cells are then going to traffic back to the organ, and those T cells are going to kill the graft. And here you can also see T cells sort of infiltrating into an organ and leading to some destruction. This is going to take longer than that couple weeks that we saw with hyperacute rejection. You're going to need at least a couple weeks to first prime the T cells, and then the T cells are going to need some time to do some damage. So this is going to happen realistically over the first few months following the transplant. So there is going to, at first, be a sensitization phase where whatever the antigen, the alloantigen here, whatever the alloantigen is, it's going to make its way to the lymph node to actually activate some T cells. So first we've got to get antigen into the lymph node to turn on T cells. And once those T cells are on, those T cells are going to have to migrate back to the organ and start to kill that organ. This gets us to kind of this other major issue of immunology, which has to do with sort of how we understand this process. Basically what we're going to be talking about here, and we're going to be talking about this for the next sort of few slides, the next bit of class, is what is the antigen? There are a couple answers to what the antigen could be. We're going to talk about kind of most common, less common. This is always a tricky piece of the semester, because what this eventually leads me to have to explain to you is that when I told you about something else earlier this semester, I oversimplified it. And that oversimplification was really useful for all of us before, and now that oversimplification we can't really have anymore. I've got to tell you the real story. So this lecture always feels funny because it feels like I have to tell you that I partially lied to you before. But I didn't. I just oversimplified. And I can even get into, like, there's stuff one can say. There's a lot one can say about this. But, so there might be a moment where you're like, whoa, 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 whoa. Yeah, you're right. So what has been realized is that one of the biggest antigens that's important here is MHC. In fact, what we're thinking about with transplantation is whether tissues are compatible. The sort of word that's often used in sort of anatomy and physiology types terms when you're thinking about tissues is histo, like histology. Think about are tissues compatible for transplant? You call that histocompatibility. You remember MHC stands for major histocompatibility complex. And the major antigen, in this case, is MHC. That's how it got major in its name. A lot of this comes up with experiments like this, where we can see situations where we might have a donor mouse of strain A giving a transplant to a recipient also of strain A, and we're going to see that graft is not rejected, it's going to take. It's also called here a syngeneic graft, syngeneic and autogeneic, or auto. Syn and auto here are both used for the same thing. You can also see a situation where we have a mismatch. So we've got mouse of strain B, recipient of strain A, fully foreign graft, fully allogeneic graft, rejected. Experiments like these are actually how mouse MHC genetics were worked out. And there was a Nobel Prize given for experiments like these. And in those experiments, people did further work to try to work out the genetics. They found things that if they had a mouse of strain B, it went into a mouse that was strain A times B, so it had that, the MHC that was on this graft was self, no rejection. Whereas if you put in skin from this mouse into another, now we've got some foreign stuff in this graft. The graft is rejected. You can also note that here we're actually looking at the hybrid, or the child of these two different parents. And so this talks about the parent to child versus child to parent. I'm not going to focus on that too much because sometimes every year people get really like worried about whether or not they're giving kidneys to their parents. And I'm like, guys, you're outbred. You're not inbred mice. Like this applies to inbred mice and childs and parents. Not outbred species, so that's why I'm kind of not focusing on it. And we also know this in people as well. So this actually shows us the importance of MHC in transplant matching in humans. And so what we're looking at here are the number of MHCs that are mismatched between the donor and the recipient. So we can imagine 0 mismatches to 6 mismatches. Here we're largely talking about class 1, because remember class 1 is on all cells, like kidney cells. So we might have 0 mismatches, or we might have 6 mismatches of MHC. And you can see the percent of the organs that actually last in our patient for 10 years. And you can see the half-life of the organ. And what you can notice is that with more MHC mismatches, you have more of a problem. And so clearly MHC matching is really important in having your transplant persist. So this is showing us this importance of MHC in transplantation. And so this tells us about how that MHC is such a major thing. So, I've shown you this before. This is the classic view of this. And this issue of MHC restriction. So, classically, when a T cell receptor is binding to MHC plus peptide, we have to have a match with the correct MHC and the correct peptide in order for that T cell to recognize and that T cell to do something. But I told you before. Now I just told you, if I give you a kidney with the wrong MHC, your T cells are going to kill it. Here's a T cell with wrong MHC, and before I told you, it didn't work. So it's a little bit oversimplification. This experiment, this is really the way that this experiment did come out. The experiment that got the Nobel Prize for this was totally clean and came out totally cleanly like this. And the reason for that is that Doherty and Zinkernagel happened to pick 2 mouse strains where this actually works perfectly. They just by luck picked 2 mouse strains where it would actually work like this. But in most cases, it doesn't actually work quite this cleanly. Because if you think about what the T cell is seeing, so here you can see what the T cell is seeing with MHC plus peptide. And you're sort of looking at the amino acids, they're color-coded here based on whether they're from the peptide or from the MHC. If we took that color coding away, would you be able to tell which amino acids were from the peptide and which were from the MHC? No. The T cell doesn't have color coding. The T cell just sees a field of amino acids. It just sees a surface. And so the T cell is really making its distinction based on how well it binds to that surface. Sometimes the surface of the MHC is just so different than what the T cell usually sees, it's not possible to have binding. Zinkernagel and Doherty picked 2 mice that had such different MHC types that there was no way this T cell could interact with this MHC because they were so structurally different. But more commonly, things look a little bit like this. So here you can see an example of the self-MHC presenting a peptide to the T cell receptor. And the T cell receptor is seeing this entire surface of MHC plus peptide. So you can see the T cell receptor is making contact with the little knobby amino acids at the end of the MHC, as well as making contacts with the square amino acids of the peptide, and that's turning the T cell on. However, if we had a different MHC that happened to have those same knobby ends, and itself had square amino acids, so now you can see there's still squares, it's just the squares are coming from the shape of the MHC, not the peptide. If the MHC happened to have square amino acids there, it could bind in that surface. And it could turn on that same T cell. You could also have a situation where that MHC has the knobby things and has one of the squares, and it matches this T cell when it happens to have a peptide with the other square. So sometimes it's sort of the combo of the MHC plus peptide can give you a reaction. So the idea here is that if two MHC types are sort of similar-ish, the T cell can recognize and think there's a foreign peptide. So here, this could be a self-peptide, but the MHC fakes the T cell out. It looks like this foreign peptide. And so this foreign MHC just looks like a foreign peptide and turns on the T cell. What I've shown you here is known as direct allorecognition. And so the thing that can happen is that sometimes the cells of the graft are presenting on whatever their MHC type is, and the T cells sort of see that and think that it is self-MHC plus peptide. They're sort of similar enough biochemically that we can have sort of that cross reaction. And so we've got some cell from the donor presenting antigen. It might be presenting it could be presenting whatever peptide. And the T cell kind of mistakes this for self-MHC presenting a foreign peptide, and the T cell kills. You can also see direct allorecognition on the left side of this. And so the idea here is that our donor cell will be presenting on its MHC. The donor cell note is yellow and its MHC is yellow. It's presenting some rando peptides to the T cells. The T cells are directly activated and they're like, oh, this cell is so bad. Right? There is one other way that someone else's MHC can be perceived by your T cells as foreign. This is known as indirect allorecognition. With indirect allorecognition, we are assuming that some cell from the graft dies. So let's imagine the situation of a kidney transplant. Why might there be some dead cells in that kidney graft?
Just from the trauma of surgery?
Yeah, from like the trauma of getting cut out of someone, the trauma of getting put into someone, of getting blood vessels, the trauma of being in a cooler between patients. Some cells are going to die. They're going to be dead cells, right? Those dead cells, when that kidney goes into you, are going to be phagocytosed by some of your cells. Your cells are going to take that dead cell and degrade it up, as one does, including degrading all the proteins in that cell. One of the proteins in that cell is MHC. Because remember, all cells have MHC plus 1. If the MHC plus 1, and that MHC plus 1 is going to, the other person, the not you, going to get chewed up into peptides, and your MHC is going to present those peptides. And since that MHC is not your MHC, that's going to be a foreign peptide. And so now, we've got your dendritic cells, your APC, recipient APC, that is presenting peptides from the donor And so now you're presenting that actual foreign peptide on your MHC. This kind of should look like the standard MHC presentation and recognition we talked about earlier in the semester. And your T cells are going to respond. You can see this here as well. We've got dead cells from our kidney that came in. We're going to have those dead, they have their yellow MHC on their surface. That cell is going to be phagocytosed. That MHC is going to get inside. It's going to get degraded into yellow peptides. Those yellow peptides can now be presented on this cell, orange MHC, and presented to the T cell. And so this is the indirect recognition of that foreign MHC. And so both of these can be an issue in turning on T cells to potentially attack that graft. What you can imagine is that these are going to take different amounts of time. And so sort of it's not like if you get a kidney transplant, you always know on like the third Tuesday is going to be the day that like rejection happens or something. There's a lot of variation here in what's going on with the rejection. And that may even come into things like what's the sort of biochemical structure of your MHC versus their MHC, and how does that mismatch actually play out biochemically? There's a lot of variation here. That's why I'm not giving you like specific timing. This rejection can happen through either direct or indirect allorecognition. So, does this make sense? Yep, Krishna.
So are these two ways that someone with different MHC could be compatible with you, or how the different MHC you could reject it?
So these are two different ways your T cells could get turned on to reject a graft from a person of different MHC.
Okay.
So if you do not have a perfect MHC match with someone else, this is what your T cells could do to that kidney.
Okay.
So there are two ways your T cells could get activated in response to that kidney. Yep.
What are the symptoms of this type of rejection?
It's really sort of loss of function of the graft. So now let's think about this. Let's imagine, how could you, if you were a transplant surgeon, how could you prevent this from happening? Yeah, Rishi.
Blood tests.
Blood tests? What kind of blood tests? Specifically, what are you doing with those blood tests?
Looking for like the correct MHC type.
You're looking for correct MHC type. So this is why we do MHC matching.
There you go.
What do you have to do for your MHC matching here? If you want to completely avoid this. Yeah, Jamie.
You have to sequence?
Okay, you might have to sequence. But note that you've got to match all the MHC types. How likely is it that you're going to find a perfect match across all MHC types? It's going to be really hard, right? To get a perfect MHC match across all MHC types because humans are so outbred. This is one of the reasons why transplantation is so hard and why patients spend so long waiting for a transplant. Because they're not just waiting for any old kidney. They're waiting for a kidney that actually is a match for them. This is a huge issue in transplantation, is trying to get those matches. It's actually very rare that we can get a perfect match because humans are so outbred. And that's why, we see things like this, where we sometimes will see, well, maybe there's one mismatch. Or in general, we try to make sure there are 3 or fewer. You want to have as few mismatches as possible, but it can be really hard to get a perfect match. So one thing you want to do here is MHC matching. So there's something else that you can do, which we're going to get into shortly. But there's also one other piece of this that we've got to think a little bit about, which is that I show you here what happens with all these different numbers of mismatches, including 0. So I show you a case where we do it. We get the perfect MHC match. You can see that it's not that 100% of the kidneys are going to survive. And honestly, there's never a situation where 100% are going to survive for a lot of reasons outside of the immunology of transplants. But you can see that those transplants are not gonna survive 100% of the time. So we need to think about what's going on in that case. What's the antigen in that case? I'm gonna sort of talk about an example I'm gonna give you right now is a little bit of a weird example, but it also is kind of one of the particularly extreme examples of this that I hope will help you understand. Okay? So let's imagine I wake up tomorrow and find out I need a kidney. And let's imagine that you guys are all very generous and Sebastian decides to give me a kidney. If that happened, and let's imagine that Sebastian and I were a perfect MHC match. I would still reject Sebastian's kidney. I picked Sebastian on purpose. Why would I reject Sebastian's kidney? It's the same reason why I would reject Rishi's kidney, Justin's kidney, or Jin's kidney. Yep.
It depends on gender.
Okay. So there's a difference here. So they're male and female. So so there's something going on with gender. What's the deal? Like, let's actually think about what the deal is here. Yeah, Rishi.
Hormones.
It's not hormones. Oh, but it's good for trying. Yeah.
Chromosomes?
Chromosomes. What's the chromosome difference?
XX and it's one.
Yeah, you guys have an extra chromosome. And that chromosome encodes a bunch of proteins, all of which are self-antigens to you and foreign antigens to me. If you look at every person in this room, every one of you has some proteins that are different than my proteins, right? That's how an outside species works. Is you all have some proteins that are not identical to mine. The most obvious example are the Y chromosome encoded proteins. But you all have some protein that is different, because you are all not the same person as me. Any of those other proteins could be an antigen that could result in me rejecting your graft. Those are all known as minor histocompatibility antigens. So here you can see the description that this book is giving is that they are polymorphic self-proteins that differ in amino acid seen in individuals. It's basically any protein between you and me that's not the same. Can be a minor histocompatibility antigen and can actually lead to rejection. This is going to largely be related, and so here you can see in the donor red protein is self, and in the recipient blue protein is self, because when we have that rejection or that mismatch, you could either imagine directly or indirectly those antigens can be recognized by T cells and can lead to death of the graft. And you can see this here as well. Here we can see a situation where we have mice with a major histocompatibility mismatch. So we've got a mouse of MHC type A. We're giving its skin to a mouse of MHC type B. We can see that that graft is rejected within about 10 days. But if we have two mice that are MHC identical but have some other polymorphism, some other difference between them, here it's the difference between yellow and orange, you can see that we still get rejection, it's just a little bit more slow. And so, again, unless you are getting a transplant from yourself, basically, or from your identical twin, there is some minor histocompatibility mismatch. Yep.
So would a female be able to donate to a male because they still have an X chromosome?
I mean, you wouldn't have to worry about the Y chromosome stuff, but you could probably have some other protein that differs in your body, so. Yeah. Yes, you don't have to worry about the Y chromosome problem there, which is the most obvious problem, but you probably have other problems. And they do not, they give male organs to females when we do transplants because you just can't match all this. You can't, there are so few organs, you can't be that picky.
Yeah.
But in that case, you might imagine this kind of problem. The final aspect of this that I want to talk about is the chronic rejection process. So chronic rejection is the final type of rejection. You can see that chronic rejection is similar to a type 3 hypersensitivity response. So based on that, what do you know about chronic rejection? Yeah, Jamie.
It's going to be antibodies, but it's going to be from the antigen as a secreted protein.
Yep, so it's going to, and so what was the other name for a type 3 hypersensitivity? Krishna?
Immune complex disease.
Immune complex disease. So here we're going to have antibodies that are the problem, and immune complexes are going to be largely our issue. So the idea here is that we're going to have antibodies that are going to be binding. Potentially, they can actually not necessarily be soluble antigen. We could have some cell surface antigen here too. We're going to have antibodies binding to the vasculature. We're going to then get some tissue damage. We also are likely going to get some immune complexes that are going to occlude the vessel. We're going to get vessel damage because of, we're going to get immune complexes, and then we're going to get inflammation because of those immune complexes, all that kind of good stuff. The sort of key thing here with chronic rejection is that these antibodies are not pre-existing antibodies. These antibodies have to be primed after transplant. So we've got to have our donor cell peptides turn on some T cells, and those T cells have to react with a B cell, and then the antibodies are made. This takes a while, and this is something that usually is going to be sort of years following transplantation. We actually don't understand a lot about chronic rejection, and the biggest reason why we don't understand a lot about chronic rejection is that for most of the time we've been doing transplantation, we had to deal with acute rejection, and transplants didn't stick around long enough to get to the chronic phase. So we're only kind of learning more about chronic rejection now, but it does seem to be more about antibodies that you primed in response to the donor antigen. The other thing that I want to talk a little bit about really briefly is bone marrow transplant. So the problem with bone marrow transplantation is that there is this additional complication in bone marrow transplantation. When we talked about solid organ transplantation, we just saw 3 different ways that a solid organ could be rejected. All of those types of rejection could be called, no one calls them this, but we could call them, host versus graft disease. The idea is that I kill the kidney, right? I'm the host, I kill the kidney that you put in me. With bone marrow transplantation, there is an additional issue that can happen, which is called graft versus host disease, where the bone marrow tries to kill you instead of you trying to kill the bone marrow. Bone marrow transplantation is done pretty commonly. I'm going to skip the next slide. Just there are a couple slides here that I think don't make a ton of sense right now, so we'll skip them. So this is sort of the general idea of bone marrow transplantation, at least historically. So with bone marrow transplantation, we usually will actually go into the bone, get marrow out. Historically, this was actually done by a needle to the hip bone to get a marrow out of the hip bone. And so we take that marrow from a person, and then we're going to actually transplant in bone marrow. In general, when we do that, we have to wipe out the immune system of the person first, either by radiation or chemotherapy, to get rid of the entire hematopoietic system of that person. So we get rid of the hematopoietic system, Imagine that is not great and is rather dangerous. We make the person empty. And then we give them bone marrow in order to reconstitute. This can happen both because someone has to get radiation and chemo as just kind of a side effect of, oops, we killed their immune system, we need to give them one back. Or we can have a situation where there's a problem with your immune system and we have to actually transplant one in. So for example, if you were born without Reg 1 or Reg 2, we would get rid of your immune cells and we transplant in some that had Reg 1 and Reg 2 so that you could actually go through the recombination. So there's sort of two different reasons why you might be doing this. And when you do this, you have something like this occurring, where we're going to take the bone marrow and put it into our patient. Bone marrow is a mixture of a lot of cell types. Bone marrow contains some red blood cells. Bone marrow contains the stem cells that are the things we actually are trying to transplant here to help you grow a new immune system. But bone marrow also contains memory T cells and memory B cells. The bone marrow, if you remember, does a lot of our B cell and T cell development. It has a lot of great cytokines to help cells live. And so it's a great place for memory cells to go and get some cytokines so they can live. So in fact, a lot of our memory cells like to live in the bone marrow, especially memory T cells. So when we transplant that bone marrow, those memory T cells will be in there. Those memory T cells will go to this person's lymph node. See, they'll go to the person's lymph node. Get to the lymph node, and they'll be like, oh my gosh, look at how many foreign things. And those T cells are going to get very much activated. Because they're going to be seeing all the foreign antigens of both the major and the minor histocompatibility complexes. Those T cells are going to proliferate a lot. And they're going to start trying to kill anything with these foreign antigens, which is kind of going to be all the things. And so those T cells are going to actually start going through the body, causing damage. You can see some of the specifics of this here, of what can happen depending on the severity. We can go from, we can have rash, up to even further liver destruction, massive GI tract destruction, all sorts of things like that, all as a result of graft versus host disease. There's some details about matching and the importance of matching that can be dealt with later, but there's one final thing I want to tell you about graft versus host disease and kind of how we deal with it. So based on what I've shown you about graft versus host disease, can you imagine what you might do to avoid it if you were a doctor who was doing all this stuff? We actually have very little graft versus host disease now.
Would it have to do with MHC taking it down?
Nope. What we do now is, after we take the bone marrow out of the patient, we actually run some purification steps and just get the stem cells. And so instead of transplanting the whole bone marrow, we just find the stem cells, the hematopoietic stem cells, and transplant the hematopoietic stem cells that have not already become T cells and B cells. So they can learn the new host as self, instead of having those T cells that are already activated. So now we basically will do hematopoietic stem cell transplantation instead of full bone marrow transplantation. One thing that's also kind of cool is we found out that some hematopoietic stem cells circulate in the blood. So now if you want to give someone a bone marrow transplant, you actually can take blood from a donor instead of having to do an injection into their hip to get marrow out. So that's another kind of perk of this, though the real perk is you avoid graft versus host. I will see you guys on Monday. I'm probably going to, the plan for next week, I think, is I've got vaccines and cancer I still need to talk about. I'm going to get rid of HIV, because I do need to also talk about some immunotherapy types of things, both for transplantation and for autoimmune. So I will organize stuff and either do immunotherapy or vaccines on Monday. And have a great weekend. I'll see you Monday.